Welcome to the part 3 of the enzyme. So on this video, I will be tackling to you the models of enzyme actions. So to understand how enzyme acts on a particular substrate, let's look on these two models of enzyme action. Let's take a look at this enzyme active site. What is an active site? It says here, an active site is a small part of an enzyme structure. This is where the catalysis takes place. So an active site is a part of the enzyme where catalysis takes place. This is a place, or this is a part of the enzyme where the substrate specifically binds. Okay? It says here also that an active site is a crevice-like location in the enzyme. Okay? So this is where the substrate binds, and this is where the catalysis or the actual catalysis takes place. In some enzyme, okay, there are more than one active sites. Okay? And of course, remember, the enzyme must be a protein that is globular. The second model is called the enzyme substrate complex. So how does this complex happen? When a substrate binds to the enzyme, we form what we call an enzyme substrate complex. Okay? What is this? This is an intermediate reaction that is formed when the substrate binds to the active site. This is the beginning, okay? This is the beginning of the transformation. For example, the enzyme here is sucrase. Okay, sucrase. Let's put C here. And the substrate is sucrose. Okay, the enzyme is globular. All right, let's assume that somewhere here is the active site. Okay, and here's the sucrose. All right, the sucrose. When sucrose binds to this active site, the complex is formed. What is the name of the complex? Sucrase-sucrose complex. Sucrose or sucrase-sucrose complex. And that's the beginning of transformation. Okay? The formation of the enzyme substrate complex is affected by orientation and proximity. What is orientation? When the substrate has similar shape with the active site, that is orientation. Okay, when the substrate has the or has similar shape with the active site, that is the orientation. Proximity is the closeness, okay, or the distance or the closeness of the substrate towards the enzyme. Okay, so the formation of the complex is affected by orientation and proximity. So this complex becomes more favorable and the reaction becomes fast if the orientation and the proximity are favorable. Okay? When when the substrate is formed, what will happen to the sucrose? The sucrose will split into two, and we will form actually a product. The substrate is converted into product. Okay? So here, again, you have a sucrose that is a substrate. When sucrose binds to the sucrase, there's a transformation that takes place in the active site. When the catalysis is over, the product, okay, the product leaves the enzyme. Okay, the product leaves the active site of the enzyme. The enzyme becomes unaffected. It does not undergo any transformation, but it's the substrate that is converted to the product. To understand more fully about the action of enzyme, let's see these two models for substrate binding. Okay, as I said earlier, when a substrate binds to the enzyme, the intermediate product is called the enzyme substrate complex. This complex illustrates that the substrate is beginning to be converted into a product. So after this ES complex, we will now have E plus P. That means the product now attaches from the enzyme. Or sorry, detaches from the enzyme. Okay? Two models of substrate binding. One is lock and key model. The other one is induce fit model. What is a lock and key? Let's assume that you have a padlock. Okay. This is not a padlock, but I would like to illustrate enzyme with a padlock. Okay. A padlock opens only if you use its specific key. Okay, so for example, you have a padlock here, and here's the opening where it receives the key, and here's the key. Okay, the groove inside this padlock is similar on the what shape, the general shape of the key. So similar to this model, lock and key model, it says here, enzyme has a predetermined shape for the active site. So for example, here's the enzyme. Okay, this is the enzyme, and this portion here, this is the active site. It says in the lock and key model, the substrate must perfectly bind to the active site because the shape of the active site resembles the shape of the substrate. Okay? Resembles the shape of the active site resembles the shape of the substrate. Of course, if the active site is, say, like this, the substrate does not fit here because they do not have the same geometrical shape. Okay? So basically it says only substrate of a specific shape can bind with the active site. That's lock and key model. The second model of substrate binding to enzyme is what we call Induce Fit Model. Earlier, we have seen that in lock and key model, in order for the binding to take place, the substrate must have the same geometric shape with the active site. However, in the induced fit, this model allows small changes to accommodate the substrate. For example, here's the enzyme, okay, and here's the substrate. Take note that they do not have the same geometrical shape. The active site and the substrate do not have the same geometrical shape. However, in induced fit model, this enzyme rearranges, okay, to see to it that the substrate can be accommodated in the active site. Okay, so that is induced fit model. So when we say you have induced fit model, okay, that's not an absolute enzyme, whereas lock and key, okay, lock and key model, that falls to this specificity, absolute specificity. It says an enzyme catalyzes a particular reaction for a particular substrate. The reason why it catalyzes only one particular substrate because it can only recognize the specific substrate. For example, the enzyme is lactase. Okay? It only recognizes lactose. All right? It cannot recognize any sugar but only lactose. Okay? Another specificity is called stereochemical specificity. So this enzyme can distinguish between different kinds of isomers. For example, L-oxidase, L-amino acid oxidase catalyzes only, or it recognizes only the L-amino acid. Okay, but not the D-amino acid, although they have the same molecular formula, but the arrangement, the spatial arrangement of atoms in space is different, so that cannot be recognized by this enzyme. Okay, so the enzyme recognizes the substrate based on the stereochemical arrangement of the enzyme. Okay, then we have group specificity. This involves structurally similar compounds that have the same functional group. So carboxypeptidase cleaves the amino acid at a time from the carboxyl of the peptide chain. So this group of enzyme recognizes the carboxyl end. Okay, so this is not this recognizes the carboxyl group. And linkage specificity, they are specific in a sense that they catalyze the reaction based on the type of linkage. For example, phosphatases, they hydrolyze or they cleave the phosphate ester bond. Okay, so these are the specificity of the enzymes.